let's move on to our final presentation this evening, which is Les Underhill. I think it's wonderful having having someone like Les with his statistical mind to assist us in making sure that the citizen science projects have meaningful application. And so Les is going to tell us about using citizen science data to develop higher level distribution maps. Les, over to you.
Thanks, Rick. Can you hear me?
Yes,
Okay,
perfect, and we
good.
can see your presentation. Yeah.
Yeah. So these are called next level distribution maps. It's the term next level is borrowed from Blasio Vuvuzello for that yeah, nothing more said. So I remind you what the discipline of statistics is really about. We see data as a signal plus noise. That's the fundamental orientation. Not many statistics courses say that, but that's the reality. And my job as a statistician is to try and extract the signal from the noise. So it's a noise elimination. So it's all the biases, all the errors that go into data. Signal plus noise. So jumping straight in, here is the bird picks map for the Cape Longclaw in South Africa, Lesotho, and Swaziland. So this is just a presence-absence map, just presence-absence. So there's no indication of how many records there are in this display. So we're told that there are 339 records from 144 grid cells, and there are 2,000 grid cells in this region. So that's actually summarized as quite pathetic, and that's true of a huge amount of the data that we've collected, even for the bird atlas itself in areas outside of South Africa. So you plot the map for a species in Nigeria or Kenya or Namibia, and it's actually very disappointing how incomplete that distribution is in relation to what we know it must look like. So so these maps are actually not very different from the published maps in the reptile atlas or the butterfly atlas, maps which have been considered absolutely amazing. So this is an example, the bush scarlet butterfly, 660 records for 173 grid cells. The orange squares are specimen records, the turquoise circles are the virtual museum records. And this is the best we can do for many species, and it's only for the birds that that we can actually produce high quality, high definition maps, thanks to people like Johan and the citizen scientists who participate in the bird atlas. It's only for birds that we can actually make really, really good data. And you look at a map like this one, and you can see the species must be occurring in all these holes in the distribution. So you know, we know what happens there, but around here, this must be sort of something like this as the pattern of the distribution. So it needs some sort of interpolation to get it going. So 339 records from 144 grid cells for the Cape Longclaw presence-absence map. This is what we do a lot of presenting of. For some purposes, this might be perfectly adequate, but it can be a problem, as we'll see later on, for other species. This is the painted lady butterfly, universal butterfly almost throughout the world. In this map, there are 4,958 records from 843 grid cells. Once again, the specimen records separated from the virtual museum records in this presentation. But when you look at the map, the impression, so this is the grid cell is colored if it has got at least one record. And if you look at this map, one sees a sort of a hole in the distribution over the northern Cape, and almost continuous distribution over the eastern half of the country. And so you'd be forgiven for thinking that this is a species that is concentrated along the coast and is absent from the interior. And you'll see later on that this probably isn't the reality. Okay. Okay, so that brings us on to what I call the observer process. So observer process is really what is causing a lot of the noise in data. So observer doesn't necessarily mean citizen scientist observer. It can be professional biologist measuring the size of eggs, and there's a little bit of error, a little bit of noise in the measurements. So signal plus noise, and we are trying to find the signal, the actual distribution of the species. That's what signal means in this context. So where does the noise come from? Well, first of all, we don't go everywhere. So there's lots of grid cells that are unvisited, and that's what is the cause of the false negatives. The species actually occurs, but it's not being recorded there. So some grid cells have vast amounts of data, thousands of records, and so species which just occur occasionally or as vagrants, they are recorded and their presence gets reported on the presence-absence map. Some grid cells have very little data, so even common species are missing. Some observers, particularly with the virtual museum, they know painted lady has been recorded in this grid cell already. They're not going to bother to record it again, so they neglect the common species. Some observers like to focus on certain groups, so some people might like to just do water birds and not all the other species. Another big bias for many groups of species is that fact that we are just not there at the right time. There are a couple of months when a dragonfly is in flight, or when a migrant bird is summertime, when the migrants are present. So here are the basic ideas for these new maps. The maps that I'll be talking about, they use every record, not just the records for the species that we are mapping as the next species to map. And there's also a fundamental assumption, which may or may not be right. If a species occurs at a place, it is likely that it occurs in the neighbourhood of that place, where neighbourhood is very vaguely defined at this point. So the strategy for filling in the gaps, interpolation, and statisticians use the word imputing for filling in gaps in data. So that strategy depends on the idea that the species occurs in the neighbourhoods of places where it actually has been recorded in the past. So in this method as I'm describing to you, the neighbourhood is defined adaptively. Adaptively is a technical statistical term in this context. So the neighbourhood of a grid cell is tiny if there's lots of data in the grid cell and its immediate area. So if there's lots of data, there's no need to do any interpolation. The data is, lots of data is available. But if there's not much data in the immediate neighborhood, then this neighborhood concept goes out in a wider and wider circle until it's got enough data. And the technicalities and the equations of that are quite complicated, but it's just a concept that we need to talk about tonight. So for the butterflies, there's one set of neighborhoods. Each grid cell has a neighborhood, the amount of data, how far you have to go to get enough data for that species. So it's not species specific, it's taxon specific. So, so moving along, here is the bird picks data, the actual coverage. And the biggest dots have, or where there's 100 or more species, and there are littler dots where there's fewer than 100 species. So, you know, once again, it's not an awful lot of data to work with. So about 120,000 records at the moment. And we all say to everybody up and down Africa, please boost this total. Further north in Africa, we're talking to Michael Brooks, and he's actually starting to put the bird picks records in as contributions to the Bird Atlas project, incidental contributions. So this top map is the one from the previous slide, which shows where the data is, and the map, new map with the yellow and purple colors, shows the reliability for the bird picks data for any grid cell. So if the color is yellow, as it is up at the top there, it means that there's not much data, and you have to go out a long way to find enough data to do the interpolation. So that's, this area corresponds to that area, and you can see these areas are the second shade. So the interpolation there is in fact not very reliable. So you can actually not only see where the species has been interpolated to occur, you can also see whether I have any faith in the interpolation I've done. So the paler shades for birds in this part of Northwest Province, over much of the Northern Cape, and over much of the Eastern Cape, of course the Karoo as well. So back to the Cape Longclaw, the algorithm looks at the data in this neighborhood of the grid cell, which might be small if there's lots of data, broad if there's only a little data, and it asks the question, is the Cape Longclaw in the neighborhood, small or large? And if yes, it says what proportion of the records are Cape Longclaw. It sorts all these proportions, and the top third are called the inner core of the distribution, the middle core, and the bottom third are called the periphery of the distributions. So it doesn't actually use the values, it actually sorts them into groups, and that way one actually deals with outliers rather well. So when I've applied this method to the Cape Longclaw data, this is the map that I get. The light shade is where I've considered the periphery to be, and the darkish purple shade is where the core of the distribution is, a third in each of these categories for what it's worth. And the birds is the only thing that we actually have the definitive distribution maps for the Bird Atlas project, but almost definitive distribution maps, because when you get to the Bird Atlas data, it looks like this. So this is Bird Atlas data for the orange-throated longclaw, The red is where it occurs, its reporting rate. The blue is where we have SABAP2 data. And it's actually quite horrifying after 13 years of SABAP2 that there are still such vast areas of the country of South Africa with so little data. So this is real consolation for countries further north, which are battling also with coverage issues. So the first thing that I have to do is to apply the interpolation algorithm to the SABAP2 data, so that I actually get a complete map of the distribution. And then because the SABAP2 data is on a pentad scale, and the bird pix data is on a quarter-degree grid cell, I then have to degrade the pentad scale map to a quarter-degree map. And this is what the map for the orange-throated longclaw looks like. So SABAP2 data, quarter-degree grid cell map. The pentad map looks much, much better than this, and actually is I think, a pretty good map of the distribution. So you put these things together, and you get this comparison. This map shows that there's almost no problems with interpolation. There's just a few areas where the SABAP2 data is a little bit thin. Otherwise, the distribution mapping can be considered reliable. So now you get the four maps. The top left is the raw atlas data, bird atlas data. Top right is the bird pix data. Is the smooth map from the SABAP2 data. Bottom left is the handful of records from the orange-throated longclaw, and the bottom right is the smooth maps for the orange-throated longclaw, Cape longclaw. So it's amazing how well the interpolation has performed, considering how little data it has to work with. So so I'm actually quite encouraged. I'm still fine-tuning the algorithm, but I think it's going in the right direction. So what happened to the painted lady when I applied this algorithm to the painted lady? So there on the left is the raw data for the painted lady, which you've seen before, where they occur, just in this format. And on the right is the distribution that comes out of this algorithm. And what jumps out at you is that it's totally different to the raw data. So now, where there's this big hole in the distribution, is now this algorithm says it's the core of the distribution. So what has happened? And the reality is that out here in the northwest, although the painted lady is frequently recorded, the reality is that it has been recorded in both grid cells. The reality is that the proportion of painted ladies is tiny. But here in the Karoo, even though there's not much data, a high proportion of the butterflies that are seen are painted ladies. So with this method, it's telling us that the core of the distribution is where the data is sparse. So that's quite unexpected and amazing result out of this approach. Now we go to Nigeria, and I've applied the algorithm to a whole bunch of Nigerian bird species. This is at pentad scale. So the left map shows the distribution of atlasing. The blue is the distribution of atlasing across Namibia. And the data is once again fairly sparse. The red dots, which show the distribution of the African pied hornbill, that map is the red obliterates a lot of the distribution, but the blue distribution. But you can still see the data is pretty thin. And this map on the right is the algorithm's guess, different shading this time of the core, inner core, the outer core, and the periphery of the distribution of the African pied hornbill. And the Nigerian Atlas team have got to evaluate this and say whether it conforms to what they understand as the distribution. So what is encouraging about this approach, I think, is that and I'm amazed to hear myself saying this, is that we don't have to get complete coverage to get good distribution maps. We can fill in some of the gaps by a smoothing algorithm. So here are my my take-home messages. First of all, the less smoothing that we have to do, the less interpolation that we have to do, the better. So interpolation is the most dangerous operation that statisticians do. We prefer not to do it. In areas which are species poor, so for the dragonflies, the Swartland, the area just north of Cape Town, and the arid parts of the country, what helps the origin a huge amount is to have lots of data for the handful of species that do occur. And this helps prevent the algorithm from interpolating other species into these into poorly covered areas. So that's quite a technical concept. Maybe I should unpack it in writing some more. It's only for birds in South Africa that we have the complete maps from SABAP2 for testing purposes. So so the algorithm is actually being tested out on South African BirdPix data compared with SABAP2, and a large part of Caris's postgraduate studies are actually involved with that. So I think this algorithm is showing promise. I'm actually quite excited about how it's working. It's still in development, but hopefully we can actually, even with quite sparse data, generate maps that are good enough to be used in, for example, field guides for dragonflies or butterflies or scorpions or what have you. So thank you very much. That's what I wanted to talk about. Thank you.
Great, thank you very much, Les. Again, I think that it's wonderful having a such a committed and dedicated statistician on looking at this data. Obviously, it's that's what you want, isn't it? To work on the different algorithms and things like that. But yeah, I think for those of us who are participating in the various projects, citizen science projects, it's nice to know that particularly in areas that are less well covered, that records that we might be submitting from these areas can play a very important role in improving these next level distribution maps. So are there any questions for Les? I haven't checked in the chat area.
Yeah, I want to say,
Steve.
So I read it, or you have it there? Okay. So it's Kate is asking. I lost the question.
The next step could be to look at correlation with habitat, vegetation, etc.
yes, I have made progress with that. So there's lots and lots of things called species distribution models, modeling techniques, which only use the data for the species that you're working with and explanatory variables like altitude and all the rest of it. And so this approach is totally orthogonal to that approach, but certainly I think I know how to incorporate those other variables into this approach. So you have to watch this space for how to do that. But if there's lots of data, as there is for the Bird Atlas project, then interpolation is actually a really good way to come up with maps. Because as soon as you use explanatory variables, you know, so if you take one of these programs and use the world, and you do the house sparrow, then the house sparrow's distribution will be put into every part of the world where conditions are suitable for house sparrows. So now you know, when you do that, you know exactly where to introduce house sparrows, that they will just take off because the conditions are the same as in their natural range. So those methods of using explanatory variables don't take into account things like geographical divides and mountains and all the rest of it. They'll move things into areas which are quite inappropriate. So it's probably a combination of the two methods which is actually the best.
Yeah, that's what I was going to ask, because you know, as well, Les knows, and maybe a few others of you know as well, the original field guides maps were based on guesswork by lepidopterists, and the pocket guide and now the new field guide and the app are all based on the SADCA data, and it's based on some extrapolations that were done by Colin Beale and Silva Misanero, where they were looking at climatic factors. And if you take the climatic factors for a known locality for a species, then you say, well, where else do those same conditions apply? And I think it wasn't as simple as that, because obviously if they'd done it that way, they would have had some of these butterflies all over the country. So I think they had some limiting factors in there as well. And I remember being absolutely gobsmacked when I looked at the original heat map. The original versions looked like heat map because they were based on percentile polygons, whereas Les's map is based on almost the opposite way around. It's on where they don't occur so much. This one was where they occur the most. They look very like the guesswork maps, but you know something, when Les started showing me these interpolation maps, they didn't look that different to what Colin and Sylvia's maps were telling us as well. There were a few of them had a couple of unexpected range extensions, but most of them were pretty. The ones Les sent me look pretty similar. Les, you want to comment on that?
Yeah, well, I would hope they would look pretty similar. So that's sort of encouragement that the method is working reasonably well.
Yeah, I mean, some of those maps of Colin and Sylvia's were based on very small data sets. You know, things like Chrysoides phosphor, for instance, where, you know, ultimately we ended up saying, well, it's likely to occur within this geographical area anywhere you've got a certain altitude and a certain rainfall. We didn't use vegetation type, which I thought would have been advisable, but they said no, that if they put too many, what are those variables you use, Les, again, the word for those additional variables that you use?
Explanatory variables.
If you use too many of them, you just end up with a fruit salad. It becomes a complete mess. You've got to tune them, as it were, to get the best picture. And that's what they did. And well, they've worked out very well. The interesting thing is, of course, I still get emails from people saying, your book says you don't get the eyed pansy in the middle of the Karoo, but I just photographed on there, so you're wrong. Oh. Then of course you have. Oh, sorry, go on.
Sorry, Steve. There's a question from Kate. With regard to butterflies, is there a connection with the distribution of the host plant species?
Well, there must be, yeah. I mean, if you think about it, if there's no host plant, then you don't get the butterfly. However, what about migratory butterflies? You know, they don't follow that rule. The pioneer Cape of White is found in lots of places where there aren't any host plants. So I think it depends on how agile the species is, which you know, how much of an influence the host plant has on. That's one of the reasons why I don't think they used host plant as an explanatory variable, because it can actually cloud the issue.
Yeah, so it was Colin Beale, the same Colin that Steve is talking about, who put me onto the idea of interpolating as the way to go. He basically said to me, if there's enough data, then the best strategy for producing maps is to in fact interpolate. And there were no really good interpolation methods around, so I tried inventing one, and I'm actually quite happy with the tool that we've got. So this map, I think, can be used as a baseline to put under a vegetation map or an altitude map, and you can see you know, it's not going to occur at all places with that altitude. So if you have the places with that altitude within my map, then you cut it down some more, and you probably end up with something which is pretty realistic. So those are the ideas that I'm exploring next. But I think for people like Sam in Nigeria and Sydney in Kenya, I think what we are learning is that we don't actually have to go to every single place. And I travel around, say, across the Karoo, and actually get quite embarrassed at the thought that I'm expecting atlases to visit every single pentad in these areas, which are all just entirely uniform, and they must have essentially the same bird species. So why not let the interpolation algorithm actually do that, rather than insist that every single grid cell gets done over and over again? So I think Sydney and Sam, I don't think you should hear me say that, but that's what I think I'm learning.
Well, can I just show you what Colin and Sylvia's map for Venice Cardui looks like? Hang on.
Painted lady.
Do Steve?
Can you see it? It's
No,
bright.
it's very bright.
It's too bright.
Yeah.
Right, hang on. Let me just.
Can you do screen share, Steve? Have you got it on your PC?
No, it's on the app. Yes.
Yeah.
There you can see.
You put closer. If you put it closer, I think the brightness, you have to put it.
If you speak as well, so
It's just
Covers the whole country.
No, in actual fact, there's something really widespread like this butterfly or like Belos or Rota. The method falls down. So I can't wait. The new version of the app is being put together. It started now, but it's only going to come out towards the end of 2021. So I'm hoping we can use Leslie's new method and we can have those interpolated maps instead of this, or a combination of the two.
Yeah. So that map on a presence-absence basis is fine, but what I'm trying to do on top of that is actually come some way to showing the inner core, the outer core, and the periphery of the distribution. And so across the eastern half of the country, it certainly is in the distribution, but it's quite rare. So it's not every second butterfly you get as a painted lady. Whereas if you're in the Karoo, the core of the distribution, as I put it, a high proportion of the butterflies you encounter are painted ladies, and that's what this interpolation method is doing.
That's an interesting.
Can I just say, I'm probably not offering anything original when I say this, but I just wanted to say how much I agree with the generic points that you made there, Les. So, and as you know, I've done a lot of work with citizen science networks, and they give you this, they yield this unique perspective into large-scale, long-term ecological processes. And I think the combination of that with sophisticated statistical analysis is tremendously important. If I look at how these things have developed historically, I think there was a time when it was quite difficult to convince people about the what you might call the rigor of some of the citizen science data. And my feeling is that we're winning this one because we're making so much progress with the stats, and because, like you point out, you don't need to have absolute certainty in the observations. You just acknowledge that the data are produced by two things. One is the process of interest, and the other is the observation process. And if you build your statistical models which model the observation process together with the process of interest, then you can get so much out of these data. I just agree with so much of what you had to say there about the principles of doing this.
Thanks, David. I appreciate that.
Yeah,
Yes.
thanks very much for those comments.
Yes.
Steve.
Remember Gerard Talavera and his paper on the painted lady and his findings. I know he used some of the data from NepiMap for that. But what fascinates me is that it shows that the butterfly is actually an arid country adapted species. It prefers to build up its numbers in places like the Sahel before it sort of jumps over the Mediterranean and invades Europe. And then it sort of washes back the other way in the other half of the year, a bit like the American monarch, but not the same thing. And I find it fascinating that those interpolation maps have shown that, in actual fact, the core is probably the drier part of the country, because that's what it's adapted. It's not a savanna butterfly. It's not a tropical forest butterfly. It's an arid, adapted species, or semi-arid adapted species. And it'd be fascinating to see from the northern part of West Africa around the Sahel what the picture looks like there. Once our West African colleagues are getting the same sort of coverage that we're getting down here, just to see what that does for the painted lady. It's amazing. I really do.
Yeah, so talking West